Good morning to everyone. I am William Farfan Rios. I'm a postdoctoral fellow at the Living Earth Collaborative in Washington University in St. Louis and the Missouri Botanical Garden. Welcome to the Andean Amazonian Forest Responses to Climate Change Symposium. Before to continue, two important reminders. First, please use the Q&A chat section on the Zoom to post your questions at any time during the presentations. Do not use the WUBA app, please. At the end, I will deliver your questions to the speakers. And the second is indicate the name to which speaker is your question, please. Today, we have a great line of speakers, and I am thrilled to introduce first Zoraida Restrepo. She's from Cold Tree Corporation, Medellin, Colombia. Then we have Jürgen Homier from University of Göttingen, Germany. Then it's me who is going to be presenting. And lastly, we'll have Nadir Palki from the Ministry of the Environment in Peru and the University of Leeds in UK.
Hello everyone, I'm going to present our research, the primera vez de analyze about the dominant Andean tree species and the thermal tolerance. In order to address this research, we had two questions. The first one is how are the patterns in reality across the Andean tree species? And the second is performance of the dominant tree species related to the thermal tolerance at the population level. To answer this question, we have focused in the tropical Andean forest. The tropical Andean forest higher than the 1,000 meter elevation, and we have the methodology to in three different levels for the calculated pattern of rarity. The first one is the abundance, the calculated abundance of the tree of the species in the Andean forest. And for that, we're using the permanent plot from different neighbor. For example, for the rainfall, Madrid, and Coltrane, different neighbor, they have the permanent plots in the Andean forest. And we calculate the relative abundance, and the relative abundance is individual, individual per plots per species. And the second is the geographic area. The geographical rank is for calculate that we're using the GB database, and we use the GB database to logland all the records for all the species, and after that we calculate one polygon or calculate the area across the region the species can have distribution. And the fair, we have the brain needs. For the brain needs, we're using the climatic variability. The climatic variability, especially with the temperature, and we're using the work clean data for the mean temperature, the annual mean temperature. The second part is the try to understand if the thermal perform can explain the dominant species or the variety pattern in the Andean forest. And for that, we need to use two different databases. The first one is the growth rate. And for the growth rate, we're using the same plot in the dominant, the abundant species, and they calculate the relative growth rate for species per plot across all the Andes. And the second database is the relative when the climatic variability for this research. In this moment, we use the temperature, and the temperature we calculate the mean temperature, the annual mean temperature in each plot. This information is necessary to use in the symmetrical Gauss function. In this symmetrical Gauss function, we need to this information in order to calculate the three different parameters. This parameter is A is equal to the relative growth rate, and B is the temperature optimum, and C is 1.5 of the braid of the curve is or similar to the braid niche. In the first result, the first one divided all the Andean forest, the tropical Andean forest, in four regions. according with the latitude and the altitude, and we had the high regions, to high region, and to lowland regions. So we find in general we had many rare species and a few dominant species, similar to the hyperdominant species in the Amazon. But one interesting thing is also the high region, the north and the south region. For the high elevation, this region need more species in order to soon the more abundant, the more dominant, the more state. So this line is the 50% of the all the state and how many species we need to accumulate for this percent. In the high elevation, we need more species in order to accumulate all the state we need for more dominance. And the second result is this is our more dominant species in all the regions. But I think it's necessary to be focused in the how which is the species can dominate, for example, in the high elevation. And the species can dominate in the high elevation is a species with or the temperature origins. You know the distribution of the origin in the Andean is some species coming on the temperature origin and some species coming in the tropical origin. And the more is all the species the high elevation is the temperature origin. You know, for example, Quercus, Weimania, Prunus, Alnus, they are the temperature the temperate origins. And some this can suggest this species is more maybe more adapting for the coldest environmental or microalgae tropical like the tropical high Andean regions. In the thermal tolerance, it can explain the dominance. The pattern is in general. We found in a negative correlation between the thermal niche break and the thermal ultimum. You know here the size of the circle is the dominance. You can see here, for example, the more dominant species have the less the geographical area and also the less temperature optimum, but have more biggest or more longest temperature break in comparison with the rare species. In another way, for example, the relative growth rate in the Andean, all the Andean forest, the species have the similar rate and the species can grow in the same way. It's not important if the species are dominant or not dominant, they can grow in the same way. So this suggests the pattern of the dominant can be explained for another mechanism. For example, it's not related when the capacity of the species can grow. It's more related, for example, with the dispersion of the usage of the habit. For example, for the variability of the climatic change, the climatic variability. So, if the how is the pattern for the dominant species and the thermal tolerances? In general, the dominant Andean tree species have less thermal optimum than the rare species and have less geographic area. Uh, Less geographic area, so suggest the dominant species have more limited distribution area than the rare species, but they have longest thermal break needs, and this is because the species, the distribution for this species is more across the temperature gradient or the temperature elevation. The species can distribution across the mountain and occupying different microhabitat and can use it, the specialized microhabitat, and can be dominant in this part. So what is our finding in general? We have Andean forest containing a few dominant species in many of rare species. Second, rare and dominant species grow in similar rate, suggesting that the dominant pattern is also affected by the dispersal or competition of the capacity to adapt to the temporal variability of climate. Where dominant species belong to temperature regions, for example, coldness or seasonal climate, and this cloud invading of the tropical Andean after the the of life colonization in the high to the adaptation of species. This species coming here more because here is the similar climate. And finally, the dominant species may be better adapted to the coldest environment and microhabitat and maybe more resistant to the climate change than the rare species. Many thanks, and if you have some questions, let me know. Thank you.
My name is Jürgen Homer and I'm going to present you a study conducted by Selene Baez, Belen Fadrike, Ken Feely and myself. Understanding tree trait distribution along topographic gradients and through time provides insight into the processes that will shape future community composition and ecosystem functioning. In montane environments, complex topography affects critical aspects of forest composition, yet its role in determining tree trait composition, community climate indices, and responses to changing environmental conditions is not fully understood. This is an image of our study area showing the rock topography in the San Francisco Reserve in southern Ecuador. In our study, we did a repeated inventory of 18 permanent forest plots of 400 square meters each. The study investigated how functional trait composition and community climate indices vary for the tree community as a whole and for its demographic components in a topographically complex Andean forest over 8 years. We hypothesized that climate change had favored tree species with more acquisitive traits and higher thermal optima, shifting the floristic and functional tree composition. We calculated the topographic position index of the permanent plots. This schematic sketch shows slope positions and their respective TPI from most negative values at valley bottoms to the most positive values at ridge tops. Our 18 study plots are located in a narrow altitudinal belt between 1900 and 2100 meters above sea level and cover a wide range of topographic position indices. We used 8-year tree census data of the 18 monitoring plots. To test our hypothesis, we evaluated changes in floristic composition, community-weighted means for 10 functional traits, and in addition community temperature and precipitation indices as a function of topography and time. We compared the community-weighted means for the whole community and then also for the 3 basic demographic components. These are the newly recruited trees, it is the growth of the surviving trees, and the third component are the trees that died during the time from 2008 to 2016. We calculated basal area of the 3 components following the method of Muscarella et al. 2017. We did that for our 10 functional traits and also for the temperature index and for the precipitation index. As shown in the above figure, we expected shifts in the community-weighted means of the traits from the first to the second inventory and also difference between the demographic components. Species climate indices were calculated after the method published in Fadrike et al. 2018. we identified a strong influence of topography on functional tree composition, such that communities at lower topographic position, that means with lower TPI values, were dominated by acquisitive species compared to sites at upper positions dominated by more conservative species. A response that was possibly mediated by soil conditions and hydrology. In the figures you see the strong decrease of the leaf nutrients nitrogen and phosphorus and of specific leaf area with DPI. At the same time, leaves are tougher and stems have higher wood-specific gravity. Functional composition remained stable through time, and we found no evidence for trait-based functional responses to environmental change. We found also a strong influence of topography on tree species climatic optima, such that communities at lower topographic positions with lower TPI values were dominated by species adapted to both warmer and wetter conditions compared to sites at upper positions dominated by cold-adapted species. A response possibly caused by differing soil hydrology and microclimate. Our findings confirm that fine-scale environmental conditions are a critical structuring factor of tree communities in tropical montane forests, and suggest that these species-rich communities are relatively stable against slow environmental warming. This study highlights the need to explore how diverse aspects of community trait composition vary in tropical montane forests, and also to investigate thresholds of forest responses to environmental change. Thank you for your attention.
Hello, everybody. I'm William Farfan-Rios. I'm a tropical forest ecologist, and I am happy to share some preliminary results of my ongoing project in this ATBC virtual symposium. Today, I will be talking about temporary changes in species diversity across the Amazonian and Andean forests. This ongoing project is a collaborative effort between several principal researchers from different countries in Europe, United States, and South America. Thank you all for making available this valuable data set. And also, thanks to these more than 150 researchers for the effort and sweat in obtaining this massive data set, which together with the field assistants, my deep gratitude to the more than 1,000 people in the field. And personally, I spent several years working with these enthusiastic young students in the southern Peruvian plots. My gratitude to them as well. Large-scale human activity like deforestation and habitat loss and current climate change are causing significant changes in spatial and temporal patterns in species distribution. And there is evidence of biodiversity erosion from local to global scales. Despite growing evidence that biodiversity is declining due to anthropogenic forces and climate change, intense debate persists as to whether plant communities are gaining or losing species from local to global spatial scales. In this review, Valen shows that temporal trend in plant diversity at local scales is close to zero. In other words, diversity is not increasing or decreasing because of the interplay of all these different drivers of land use, habitat fragmentation, and climate warming. At the regional scale, diversity increases due to colonization, and at the global scales, plant diversity is likely to decline. But some researchers argue these results because of the poor data on time series and because there was not a good representation of tropical forest sites. And that's a caveat because tropical forests are highly vulnerable to anthropogenic forces and climate change. There is growing evidence of plant responses to climate change. For example, changes in species composition due to the thermophilization process, where cold-adapted species are replaced by warm-adapted species. Here in this study led by Fadrique, across the Andean forest from Colombia to Argentina, we showed an increase in the abundance of warm-adapted species, represented here with the red circles. If we add the Amazonian forest, we found that thermophilization in the Andes is positive and highly variable, while in the Amazon is close to zero. In other words, no thermophilization and no change in species composition. There is also evidence in forest responses to change in precipitation. Here, Esquivel et al. Showed that the dry season in the Amazon is becoming more intense since 1985, represented with the red arrows, and these events are increasing the abundance of dry affiliated taxa, potentially changing the species composition in the lowlands. Despite this growing evidence that tropical plant communities are shifting in species composition and their distributional range in response to climate change, studies on temporal changes in tree species diversity across the tropics are lacking, which led to this fundamental question. Is tree diversity in the Amazonian and Andean forests changing over time? To answer this question, first I will introduce you to the Andes and Amazon elevational gradient. Then I will show you whether diversity of tree communities in the Amazonian and Andean forests are changing, potentially in response to changing environmental conditions. Testing the lowland biotic attrition hypothesis, which is the loss of local diversity where the number of species are emigrating or going locally extinct in warm climates. And also testing the highland enrichment hypothesis, which is the increase of number of species in the cold climates. Thanks to the wonderful collaborators, we compiled 184 permanent plots in old-growth forests of the Western Amazon and Eastern Andes. I want to point that during the field campaigns, we discovered a new tree genus named in honor after the Inca Empire, Incadendron, and this is just an example of how diverse are these forests and how little we know about the nature. The data that you see here are extracted from the water climate at 3 arc seconds. Here we have temperature against elevation. Each circle represents to each plot site. And the dashed line here is the arbitrary division between the Amazonian and Andean forests, and you will see that in the next slides. You see that temperature here strongly correlates with elevation, which is well known, and precipitation correlates with elevation as well, but it seems to be constant up to 1,500 meters, and then linearly decreases up the tree line. I also want to point that along the gradient, we observe the cloud-based formation between 1,000 and 1,500 meters. This cloud base is the lower limit of the cloud immersion zone in Eastern Andes. So then we have the cloud immersion zone, and then the tree line, which is the transition zone of the forest and Andean grasslands. Now we see whether species richness in the Amazon and Andean forests are changing or not. So this is the forest inventory data. So the plots were located in the old growth forest. For each plot, we have more or equal to 2 censuses in 40 years. Each plot were revisited every 2 to 10 years, and all life forms were included above 10 centimeters in DBH. So we have almost 500,000 of stems, over 4,000 of different species. To calculate the change in species richness, first we exclude all the identified species, and then we use the taxonomic name resolution service to standardize the names. And the observed change in richness were calculated as a difference in the number of species present in the plot at the second census minus those in the first census. And to account for the variation in species richness and the length of the interval between censuses, we also calculate standardized richness change, RC here, by dividing this difference by the richness of the first census and then by the corresponding census interval length in years. Finally, the trend in richness change was calculated for each plot at the slope of the linear regression between the standardized richness change and the census year. And this is what we found. Here, the white axis represents the standardized richness change, and each circle represents 1 plot. And the red circles indicates the increase in species richness, and the blue circles indicates the loss of number of species. Overall, we found 65 plots with positive richness change and 119 plots with negative richness change. And the overall plots with negative richness change was more than expected under the null expectation of equal proportion of plots with positive and negative richness change. and the mean standardized richness change was negative and did differ significantly from zero, indicating loss of species across the plots, as you see here in this histogram. We didn't find a significant relationship of richness change with increasing elevation, but we observed a slight increase as elevation increases. However, if we look just the Amazonian plots, the richness change average was negative and significantly different from zero, supporting this biotic attrition. But we didn't find evidence of enrichments in the highlands, where the average in richness change was negative as well, rejecting the enrichment hypothesis. Something to highlight is that around the cloud base, we observe significant species loss, and around the tree line, plots seems to be increasing in the number of species. We also didn't find a significant relationship of richness change with mean annual temperature. But again, there is a tendency of losing species in warm climes and increasing the number of species in cold climes. While with precipitation, there was no relationship at all. We also observed scale effects in species richness change. At the regional scale, I show you that the lowland warm climes are significantly losing species, but also the highland plots or the highland cold climes. However, at local scales, we observe different patterns, for example, here in the central Andes of Bolivia and here in Colombia, where the pattern is clear. So the lowlands are losing species and the highlands are increasing the number of species, and this trend is significant. In summary, we observe an overall decline in richness across the Amazonian and Andean forests, and across the elevational gradient, there is a tendency of losing species richness in warm climes and increasing species richness in cold climes, with high losses in the cloud-based zone. At the regional scale, the mean negative richness change in the lowlands suggests and support the biotic attrition hypothesis, and the mean negative richness change in the highlands reject the enrichment hypothesis. At the local scale, there is evidence of biotic attrition in the lowlands and enrichment in the highlands for some sites, like in Colombia. Thank you.
Hi everyone, I'm Nadir Palki and I will present a work that calls Resilience of Carbon Stocks of Andean Forests to Shift in Composition due to Climate Change. The Andes Amazon region contains some of the most biodiverse and carbon-dense forests, where it's possible to find a wide range of type of forests from lowland to Andean tree line. In this figure, we gather recent forest studies in this region. As we can see, forests have experienced substantial changes in composition, structure, and dynamics for different environmental drivers. From these studies, we know that variation in species composition is likely to strongly influence the ability of forests to resist and recover from external perturbations. For example, the impact of changes in temperature and rainfall, and also for extreme events such as droughts and floods on ecosystems, may be moderated if compositional changes allow species adapted to these new conditions. This new arrangement of species may enhance, maintain, or reduce ecosystem processes. The links between tree mortality, recruitment, and growth, and compositional change and overall ecosystem structure are poorly understood in this region. So we use this framework to hypothesize three possible scenarios of how Andes Amazon forest resilience could be affected by compositional changes based on recent tropical studies. First, compositional change might be strongly linked to maintain biomass if it's driven by changes in tree recruitment and growth. Second, compositional change might be associated with ecosystem breakdown and biomass loss if both are driven by increased immortality. And third, while compositional change might be driven by higher mortality of some taxa, forests may still maintain or gain biomass if the growth of other taxa has simultaneously been enhanced by other processes. So to test that, these three hypotheses, we used 94 multicensus plots across Peru. 60 of them were located in lowland forests and 34 in mountain forests. And they have monitored across 2 decades overall. And use also single census plots, 219 single census plots to use as methods. It's important to notice that single census plots that are these circles are located in similar places that multicensus plots. Also, it's important to highlight that in this region we encompass 2 replicated transects, altitudinal transects, to address or explore spatial patterns. The Yanachaga transect is the first and has 19 plots. And in the south, we have Manu transect that has 24 plots. We use the thermophilization rate to analyze changes in functional composition of community over time. This rate is based on changes in community temperature index over time. So overall, the phenomenon of thermophilization is when certain community changes its composition towards having greater relative abundance of species from lower and warmer elevations. To do that, as a method, we merge 2 sources of data. The first one, we use these 1 census plots, and then we merge with GBIF data to then calculate the temperature per each occurrence. And after all these processes, we get the optimum temperature for each genus. And then we allocate these values for each multicensus plots to then get the thermophilization rate. Results. As you can see in this first graphic, there is a strong evidence for thermally associated compositional change across all plots. There is a peak thermophilization rate at mid elevation between 1000 to 2000. And there was a similar trend, a quadratic trend between thermophilization and temperature also. It's important to notice that the same trend is we found the same trend in these 2 transects, Manu and Yanachaga. But the question is, what is driving these changes in composition? The thermophilization rate could be affected by tree growth, recruitment, and mortality. We compare between drivers and thermophilization rates to show which processes is controlling this variation in this rate. At mid-elevation between 1000 to 2000 meters, the thermophilization rate are reaching a peak. Meanwhile, the CTI loss is going to the lowest point. So that suggests that the variation of thermophilization rates are driven by trends in mortality. If we compare the thermophilization rate that is in green with the CTA gain, there is no trend. But we also wanted to know which genera are changing in abundance at mid-elevation. So the role of selective mortality in driving patterns of thermophilization was particularly apparent within mid-elevation forests. For these sites, the high thermophilization rate was driven by changes in the abundance of 4 coal-affiliated genera, in particular Ediosmum, Cyathea, Clusia, and Heliocarpus, which all strongly declined in abundance at this elevation and caused the overall rise of CTI values. Across all plots, the rate of AGV change was positive. In other words, there was an overall gain in AGV. Both mountain and lowland forest plots have accumulated similar amounts of carbon. Overall, the rate of AGV change was not associated with variation in the thermophilization rate among sites. So overall, what is happening is that at mid-elevation forests are the most susceptible to environmental changes in Andes Amazon region of Peru. This rearrangement of species is related to trends in mortality of particular genera that are coal-affiliated. For mid-elevation forests show the highest rates of thermophilization and the highest rates of AGV change. This increase in AGV at mid-elevation sites is therefore not associated with a substantial shifts in growth of warmer taxa affiliated. Thanks for your attention.
Fantastic presentations. Thank you to the speakers. Hopefully these new findings will help us to understand better this pressing question of how tropical forests are responding to climate change. We have questions here in the Q&A, and also please to the assistants, if you have more questions, post it in the Q&A chat. So we have here one question. So this is the first question from Paola Cepeda. She says, thank you all for your presentations. This question goes to William. Positive richness change in Andes and Amazon could be due to the introduction of invasive and non-native species. As a mean, we showed that there was a negative richness change overall in the Andes and the Amazon, but for some plots they were positive, you're right. However, I have to say that I'm not aware that we have invasive species in the data set, because those plots are located in the old growth forests, like almost untouched, where are in the protected areas. So it seems that we don't have invasive species on our plots. So, but that's one hypothesis that need to be tested, is like the increase of invasive species are increasing the richness of some sites, but I think that's not the case in this study. We have another question, and seems that was responded from Jürgen, but just in case, Jürgen, you want to add something more. The question says from Emilio Villanova, great work. You mentioned that no changes in community weighting means of traits were found over time. So I was wondering if these 8 years, there were other changes that can be highlighted, for example, climate, high mortality.
First, thank you, William, for managing this symposium, and then Emilio, thank you for your question. As I mentioned in my answer, there are some slight changes in species composition, but there's no significant change in tree diversity. And regarding the climate, the area is relatively stable. There is a slight climate warming. It's about 0.13 degrees Celsius per decade of warming. And the amount of precipitation is not changing. So probably the area is quite stable in climate, and that's the reason that we know that we did not found a strong effect on functional composition of the trees.
Thanks, great. This question goes to Zoraida. So you found that abundant species have a wider and better thermal niche in the Andes. Did you see any difference if you go in the bands from like lower mountains, mid, and then high mountains between the abundance and the niche breadth?
William, I'm not sure, maybe yes, because now is the preliminary analysis and this is the general pattern. The braveness is more longer than the rarity species. And in general, all the species, the abundant species have this pattern in comparison with the rarity species. I think it's possible the abundance is changing across the altitude and maybe it's different the pattern, especially for the species is more in the high elevation. For example, similar for the naive Clusia, I think this genus is quite specific for the high elevation, and maybe it's possible that pattern. I think, but now it's not ready this analysis, and maybe I can do that.
Great. Here's another question from Miguel Chavez. He says, to all speakers, great presentations. Do you think that biotic interactions, pathogens, mycorrhizas, herbivores, pollinators are important determining the distribution of the species in the Andes and Amazon? If so, how will that change the current understanding of species distribution and the responses to climate change? Who wants to start?
I can start.
Perfect.
I think of course, I think the pattern for the abundance in the Andes is no relation only with the climate, on the variability for the climate. And mycorrhizas, I'm sure this is quite a strong correlation with the abundance. The problem is we know now the specific the mycorrhizas for the species, and the pattern is more across the species and not the genus, is more specific.
Jürgen, you want to add something?
Okay, thank you, Miguel, for your interesting question. I think you're right that biotic interactions are so important. We still don't understand most of them. I can only mention one example of our recent studies. We do a fertilization experiment for more than 10 years. And what we found was that the composition of mycorrhizal fungi was changed after nutrient addition, and that should have also effects on trees and tree species composition, but probably it needs more time. So our studies, most of our studies are only for 2 or 3 years, and we need decades to understand or to see even the changes in the composition of species. So we need more time to find changes and to understand the biotic interactions which are essential for species composition.
Nadir, you want to add something to the question? Maybe no, I can add something. Yes, Miguel, thanks for the question. I agree with Zoraida and Jürgen. So biotic interactions indeed are really important, and they are little neglected to study how they are shaping the distributions. A good example is that Paul find findings in the lowlands, in the white sands, in the Amazonian, how these biotic interactions shape the distribution of those species in the white sands and also in the clay soils with the translocation experiment. Indeed, that's going to be happening too in the Andes, but there is not empirical evidence. However, I know that in some plots in Colombia and also in this elevational transect in Peru, in Manu, we have those, there are those translocation experiments where the researchers move the seedlings outside of the ranges and in the ranges. See, Zoraida too is part of this project, to see exactly how those biotic interactions plus abiotic interactions are shaping the distributions of those species. So this is an ongoing project, and stay tuned for the results. I'm sure maybe in the next ATBC meeting, people will start presenting this really interesting data, and we will be able to answer your question. Great, so Here's another question. This is from Javier Jaros Carrion. Oh, Xavier Jaro Carrion, sorry. Hey, William, great study. Just a clarifying question. Does your study assume that species richness will be stable over time in the absence of changes in temperature and precipitation driven by climate change? I'm just curious how, if needed, natural species richness turnover could be separated from those driven by climate change. Yes, so Javier, the null hypothesis is going to be like no change over time. However, in reality, we know that temperature is increasing. And that hypothesis was, I think, for Cowell, maybe before. He did this study in Costa Rica, in Volcan La Barbara, elevation of Transect, I think, where he found like lowland species, lowland warm systems are losing species due warming, to climate change. And those species supposed to be immigrating upslope, increasing the richness of the mountain species. So that's the hypothesis that we tested. And we found that there is none enrichment in the mountains. So the Loon hypothesis will say no changes, but we're observing negative changes. The next step for this study will be to see that relationship with the rate of change in temperature, and also to test this empirical data against null models to actually see those differences or not, how significant are. Thanks for the question. Here we have some more comments. If you have more questions, please post it in the Q&A. There is one comment from Emilio to Zoraida. That was great. So he said, great presentation. Just quickly to let you know that we can offer data for some plots in the Venezuelan Andes to expand this work further north. So that was a nice thing. So you have now plots from Venezuela, Zoraida, available to include to your study. That was great.
Oscar, thank you. Oscar, in this moment it's possible if Emilio can share with me, Oscar, thank you.
Yes. Also, the speakers, if they have another question for the other speakers, we can do it live. No, I can start. Jorgen, so you were studying these changes in functional traits as a response, this temporal response of the climate change. So based in your experience, and we know that it's really difficult to get different functional traits of the species. So which traits the people should be looking to see those temporal changes over time? Can you suggest and elaborate a little bit more about that, please?
That's a really good question, because normally all of us, we start with the simple traits, which are easy to measure and where you can take samples of replicate trees. And so you start to collect leaves or do wood cores, and that's the commonly used traits. But I think in the future, we should maybe explore a little bit roots, for example. We started some years ago with fine roots, and because we think the relation of trees to their soil and to soil properties is very important. And so we thought, if we look at the root traits, we can find or can get more hints to this about this relation. But if we think in changes during time, so there are the traits which are related to fertility or nutrient availability. And the easiest ones are foliar nutrients, which are changing with nutrient availability, and you could expect changes and you will find them. We did that in our nutrient manipulation experiment, and we find in most species an increase in foliar nutrients. So that's maybe a good trait if you look at changes in nutrient deposition, for example. But for warming, it's a little bit more complicated, because you have to think in all the effects on tree physiology by warming. So they spend more water, transpiration will be higher, but that should have effects on leaves. Maybe leaf size is affected, specific leaf area is affected, but it's not that easy. It's not that easy to, yeah. I would need a half an hour to explain it. But I think leaf traits are interesting, because the good thing is each year there's more data available for tropical trees. Maybe you have been in that symposium on Wednesday about functional traits in the Andes. And there are each year more initiatives, more people working in these forests. And so yeah, in some years we will get a better idea which are the most important traits to sample, I think.
Indeed, it's really important, to keep building databases. And yes, this initiative of building this database of functional traits are working well, and seems that they're increasing the number of traits for all the sites in the Andes, especially where we have a really big gap in information. Here we have another question. This is from Roxana Patricia Araujo Liaga. Thank you, Patricia, for staying tuned. For the general audience, could you review a bit more about these recruits and why to include them in this modeling of a response is important? And what exactly is a recruit within population dynamics? Also to highlight that not many people knows how to identify species at the level of recruit. I think very valuable. I will let you start, Jorgen.
That's a very important comment, because if we say recruits, normally I think most of our forest inventories start at 10 centimeters diameter. Some start at 5 centimeters diameters. So we are not talking about seedlings. Normally what we mean is trees reaching this threshold of 5 or 10 centimeters. And that means that is a size where it's normally a bit easier to identify species. So it's still difficult, but easier than trying to identify seedlings. So if we're talking about, I think it's the same, William, for your data, of Zoraida or Nadia for your data. Probably most of you are working with this 10 centimeter threshold, or isn't it?
Do you have any opinions, Zoraida?
Yeah, I agree with Jürgen. It's difficult to identify the recruit, but in general, I think with the permanent plot, we try to have all the classification for the species is the diameter is the 10 diameter, and in general that is the tree, not the seedling. And I think in general, all the plot try to have the good classification for the species, and for the reason now we can use the database for this question.
Yes, Nadir, do you have any insights?
Yeah. Hi everyone. Sorry, my internet connection was off. Yeah. I agree with your opinion. It depends. Us in our case, we use the 10 centimeters diameter. So if we call recruits if is 10 or more than 10 centimeters. So it depends what threshold we use, right? Thank you.
Yes, and just to add that's a really good question about the recruits. It's ID done are really crucial because, like, for example, for studies like Nadir, we want to know which species are growing more, which species are recruiting more in the plot, so we can see how the species composition will change then in the future. For example, for Nadir's studies, like you will get thermophilic species, this warm adapted species, or you will get this cold adapted species, and then you will see the changes or not. So the recruits are the key in the study, but also which trees are dying, like Jorgen was showing. So we need to find the balance and also keep working ID those trees, and that's a humongous work. Here we have another question. This is from Sebastian Tello. To everyone, do you predict that if we were studying seedling communities, these temporal changes in traits, diversity, and composition will be stronger and more consistent? Jorgen?
That's a good idea. I think probably we would find stronger effects in the seedling communities because they are younger, and so they should be more affected. Those species composition in the seedlings should be more affected by climate change. because if you think in this, again, in this 5 or 10 centimeter threshold, the trees at that diameter already can have several decades, so up to 50 years or more in some cases. And so, but the seedlings, I think in a species-rich tropical forest, it's not possible to identify all seedlings. You need a lot of local knowledge. If you work there some 10 or 20 years, you can do it. Or with experience like William has, you can do it. But normally it's not possible. And a lot of work to identify all these seedlings because there's a high turnover. We did some seedling studies for several years, and you see from one year to another, there's a very high mortality. Almost half of the individuals is dying, and then depends on the specific year. In some years there is mass fruiting of some species, and then you get high abundance of several species, and then the next year it's another species. So in general, you should expect a stronger effect, or even compared to mature trees, but it's difficult to identify the seedling.
Thank you, Jürgen. I'm sure Zoraida and Nadir has like more insights about the seedling and me myself. So we have one more minute and I will close the symposium because the time is clicking. So in this last minute, first, I want to thank first the Zoom Tech, Patricia. Thank you, Patricia. And also Ivan Ortiz Rodriguez and the ATBC organizers for this great work in this meeting. Second, many thanks to the speakers to share their research and to the audience for their questions. If you have further questions, please use WOA to keep interacting and keep enjoying the last day of the meeting. And please, let's stay in touch. If you have more permanent plots, you can contact Zoraida, Jürgen, or Nadir. Thank you so much for this great symposium. And just the last thing is, I wanna say again, thank you to the speakers because they are in different stages of the professional careers and they were able to make it to this symposium. Really appreciate your time and your research, for sharing your research. Thank you so much.
Thank you, William. Thank you.
Thanks a lot, William.
Thank you, everyone.
Thank you.